Welcome to Wildcat Science with Mrs. Haugo. This is Human Biology, Chapter 3, Part 1. Our unit is still the body as a whole, and we are delving into cells and their functions. So to start out the chapter, we're going to figure out about heart disease from a cell's perspective and what is wrong with Jim. We'll jump into that during class. Also, we're going to do a quick review of microscopes. Please write this down. Scientists use microscopes to study cells. And then, just paying attention here, you don't have to write it down. The compound light microscope is a type of microscope we use. And then transmission electron microscope is, I'm going to pull these all up so we can see the page. On page 38, there's some good examples, some good pictures for the examples. And in A, you can see some villi or cilia through a light microscope like we could. And then a transmission electron microscope creates a picture of it using electrons, so you can get a much bigger view. And then in C, they have a scanning electron microscope, and those also use electrons, and they do the outside, so you can see what the cilia inside your throat would look like. So looking at checkpoints already, what characteristics of life does a cell show? And name 3 types of microscopes. So we studied cells quite a bit in biology, so you should know the main parts of a cell: the plasma membrane, the nucleus, and then the cytoplasm in the middle, and all of the organelles inside there. So let's delve into the plasma membrane. Some is review, some is new. What does it do? Well, it protects the cells, it's involved in communication, and it regulates what enters and leaves the cell. The components of a plasma membrane are the lipid bilayer, which is that phospholipid. Cholesterol, which we talked about before, helps stabilize the membrane, and it's also another lipid. And carbohydrates, so glycoproteins, glycogen and proteins, and glycolipids, glycogen and lipids. Those do a lot with communication. And then also there's a bunch of proteins. If we look on page 40, there's a nice picture, figure 3.3, of the plasma membrane and all that's going on. Sometimes we just call it like a little circle on the outside of a cell, but there is a lot of busyness going on in the plasma membrane. Those proteins are really integral. So let's talk about what they are, and there's a nice table on page 41. Sometimes there's channel proteins that determine what can go in and out of the plasma membrane. There are transporters if certain substances need transporting in and out, sometimes glucose. Receptors for a certain substance, kind of like your neurotransmitters in your nerve cells. Enzymes, which we know are important for catalyzing chemical reactions. And then there's linkers that link maybe one cell to another cell. And then cell identity markers, particularly important in your immune system. So you're like, this is me, don't kill me, white blood cell. Like I said, on page 41 was a nice table of those. So the membrane has a lot of potential. What do we mean by that? So when we're talking about membrane potential, we're talking about electrical potential. So there's a difference in electrical voltage on each side of the plasma membrane. Just like there's a difference in electrical voltage on the negative and the positive side of a battery, and when you connect them, you can get the electrons to move from one side to the other. Well, if you look on page 42, there is a difference in charge from one side of the plasma membrane to the other. And so sometimes a channel will open up and it'll allow the ions to flow, kind of like electricity, and then your cell can use that so that electric difference provides power to do work like generating ATP in the mitochondria. and that's very much like how a battery works. Or for cell communications, like neurons sending signals, and we'll talk about that when we get into the nervous system. So, the nucleus heading inwards, that is where the DNA is, and that the nucleus controls what happens in a cell. It's also involved in protein synthesis, and we'll talk more about that in chapter 3 later on, and involved in mitosis, and again we'll talk more about that. So in between the plasma membrane and the nucleus is the cytoplasm, all the good stuff in the middle. Alright, so the cytoplasm is the part of the cell from the nuclear membrane all the way to the plasma membrane on the edge. The cytosol is the liquid, and then it also includes all of the organelles. And organelles, we learned about them in biology. One of them is the endoplasmic reticulum. There's smooth ER and rough ER. And the question for you is, what is the function of the endoplasmic reticulum? If you said something like the rough ER sorts proteins, the smooth ER aids in lipid synthesis, you would be correct. So what I want you to do is definitely write the name of the organelle, and then if you know the function, you don't have to write it. If you knew it, if you don't know the function, you should write it out. Okay, ribosomes, what are their function? So if you know that they manufacture proteins, you're good to go. Otherwise, please write that down. Also, the mitochondria, what is the function? You said it converts energy from nutrients into ATP. You're good to go. Otherwise, write it down. The Golgi apparatus, what's its function? If you said that it sorts proteins and then prepares them for transport, you would be correct. Lysosomes, what do? They digest unwanted substances. So if you knew that, you don't have to write it down, otherwise please do so. A vesicle, what does that do? It will store and transport substances. And finally, centrioles. Their function is to separate chromosomes during mitosis, and meiosis if you're a sex cell. Let's talk a little bit more about cell structure and function. So, cell structure depends on its function, and look in your book on page 44, figure 3.5. So, a red blood cell, what can you tell me about its structure? And then what can you tell me about its function? So, if you said that it is kind of like an oval cell, an oval donut-shaped cell, and that its function is to carry lots of hemoglobin and oxygen, you would be correct. So basically it's just chock full of hemoglobin, which carries oxygen, and that flows in the blood. So its function is related to its structure. Okay, a nerve cell. What does a nerve cell look like? And what is its function? If you said that it has some branching appendages, and then a cell, and then a long branching appendage, for its structure, you would be right. And then its function is to receive messages and to send messages. Finally, a muscle cell. What is its structure and what is its function? So if you said its structure is long, thin cells, and then the function is to contract and then relax in order to make the muscle contract and relax, you would be correct. So if you knew these, you don't have to take notes. You should write down, red blood cell, nerve cell, muscle cell, structure, function. Otherwise, if you were lost on it a little bit, you'll want to take some notes on what we talked about on their structure and function. And you can go back and do that. Okay, cell structure, checkpoints, how are we doing? 3.3 on page 44. Question is, what is the main substance of the plasma membrane, and what are the 3 types of materials found within the membrane? You should be able to answer that question. 3.4, what is the membrane potential, and what types of substances maintain a membrane potential? 3.5, what are cell organelles? 3.6, what is the nucleus called? Why is it called the cell's control center? And 3.7, what are the 2 types of organelles used for movements, and what do they look like? So let's talk about the movement of substances across the plasma membrane. So starting out, the plasma membrane is semi-permeable. Perhaps you remember this term from biology. That means some substances can go through it, but not all can. And if you remember, we did a lab where we had dialysis tubing, and then we had like glucose and sucrose and iodine and starch. And the dialysis tubing, the glucose and could go through, but not the sucrose or the starch, but the iodine also could. And that it depended a lot on size. It also depends on solubility, if it's dissolvable in water or in lipids, like fats, and its charge, if it's neutral, positive, or negative. So there are a couple different types of movement. You don't need to write this down, but we will go over these: diffusion, facilitated diffusion, osmosis, filtration, active transport, and bulk transport or vesicular transport. Let's start with the types of movement that do not require energy. They will just happen naturally. One of the most important ones is osmosis, and osmosis is the movement of water. And things naturally move from high to low concentration. So if you are high to low water, then that means you will be going from where there are more solutes. If there's more solutes, there's less water, and so water moves to that. And looking on page 46, there's a nice diagram of that. There's a little U-tube where there's more solutes on the right. And so if there's more solutes, then there's less water, so the water flows to the right. So another type is diffusion, and I'll move me out of the way. And that's a movement of particles from high to low concentration. That just happens naturally. And there is a nice diagram on page 45 of this little purple substance going from all in one area at the bottom of the beaker to spreading out. And oxygen does this really well, where it moves naturally from high to low concentration, which is good for us that it moves from high concentration in our lungs to low concentration in our cells where we need it. So another type is facilitated diffusion. So these still go from high to low naturally, and it doesn't take any energy, but it doesn't just slip through the plasma membrane. It needs like a little protein. It needs a little transporter to get it through. So there is a picture of that on page 45 where there are those pink proteins that are putting the orange solutes through, but it doesn't take energy. And the last type of movement that does not require energy is filtration. And this is when water and dissolved substances move across the membrane under pressure. And this is really important in your kidneys. So looking on page 46, at the bottom on figure 3.9, you can see that there is pressure on one side, so the water is moving from the right to the left. So it's moving across the membrane under pressure, even though there's more solutes outside than inside, from the right to the left. And then looking on page 311, you can see that this is happening in the kidneys, where it's under pressure, and then the water moves from the top to the bottom. So this happens in our kidneys, where the water and a bunch of dissolved substances in your water, their blood, the blood plasma, goes from the blood into the kidneys so that it can be cleaned. In our next video, we're going to talk about movements that do require energy. I will see you then.